we have seen it's a challenge for the cells to polymerize actin to start making new actin filaments. It is a challenge for cells to maintain the actin filaments that it has produced, because there are many proteins that can sever actin filaments, and they have to be balanced with the proteins that can save actin filaments. So there's a balance between actin filament breaking proteins or depolymerizing proteins and proteins that prevent these proteins from breaking actin filaments. So let's look at two of these proteins. Let me introduce you to two proteins, tropomyosin and cofilin. Actin filaments are stabilized by the binding of tropomyosin. It is an elongated protein that binds simultaneously to 7 adjacent actin subunits in one protofilament. So here's one protofilament, and as you can see, this blue thread-like structure is tropomyosin, and this tropomyosin is binding 7 subunits in a single protofilament. Binding of tropomyosin along the actin filament can prevent the filament from interacting with other proteins. Here, although it is slightly off our main subject, but I would like to mention something about muscle contraction. In muscles, for example, we have tropomyosin and another protein attached to tropomyosin called troponin. So troponin is sensitive to calcium ion levels. When nerve impulse hits the muscle cell, it results in local increase in calcium ion. We studied that when we were talking about signaling. So that increase in calcium ion basically causes the troponin to change its structure. It goes through a conformational change because troponin has a site where it can bind calcium ion. So conformational change of troponin. Troponin in turn is bound to tropomyosin. So a conformational change in troponin causes a conformational change in tropomyosin. Tropomyosin, when it changes its conformation, it results in exposing certain domains of actin filaments. These actin filaments, these domains have been hidden by tropomyosin. When these domains are exposed, the motor protein that interacts with actin, myosin, can now interact with actin filament and result in the sliding of these two filaments, which ultimately results in muscle contraction. So since we were talking about this, I thought I would mention it here. You will not be responsible as far as for muscle contraction is concerned in this module, but learning about muscle contraction in this module. But anyways, it was relevant, so I mentioned it. So we are talking about troposin and cofilin. We have talked about tropomyosin. Now let me tell you about cofilin. Cofilin is also referred to as actin depolymerizing factor. It destabilizes actin filaments. It binds to actin filaments and also free subunits. Cofilin binds along the length of the actin filament, forcing the filament to twist. This mechanical stress weakens the contacts between actin subunits in the filament, making the filament brittle and more easily severed. Cofilin makes it easier for an ADP actin subunit to dissociate from the minus end of the filament. Rate of actin filament treadmilling is limited by slow dissociation rate at the minus end. Cofilin binding causes a large increase in the rate of actin filament treadmilling. As a result, most of actin filaments inside the cell are much shorter lived than the filaments formed from pure actin in a test tube. So again, I hope you appreciate that the cytoskeleton has to be dynamic. Once you form these polymers, you have to have mechanisms to dismantle them so cells can respond to the changes in its environment. Here we have actin filament. This is ADP actin. These, after, as we know, these monomer subunits, they assemble into filament. The ATP is hydrolyzed to ADP. I will show you an animation later on. The blue is basically the ADB version of actin monomers, and the green is the cofilin. It attaches to ADP, actin filaments containing ADP, and it causes them to disassemble. And ATP hydrolysis is generally slower than the filament assembly. So the newest actin filaments in the cell still contain mostly ATP and are resistant to depolymerization by cofilin. Cofilin, therefore, efficiently dismantles older actin filaments. this results in basically this causes a huge turnover for actin, so cells can reorganize their cytoskeletal array. Also, I would like to mention here that troponin, a specific cardiac form of protein, this troponin circulating in blood has been shown to be a biomarker for myocardial infarction or death of heart tissue. So it is an important marker. If it is detected in someone's blood, it basically suggests that that person may have had heart injury or the heart may have suffered some damage.